Today is about transcription and translation a gene. Before we start talking about transcription and translations, here are some information about DNA. Here are some information about transcription and translation. Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. So transcription is DNA to mRNA. And during translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. So translations is mRNA to proteins. These proteins perform the tasks that make living things function. The flow genetic information from DNA to mRNA to proteins is central to life. So DNA to mRNA to proteins. Transcription, in summary, is a molecular machine or RNA polymerase attached to a gene and makes a messenger RNA or mRNA copy. Here are steps of the transcription. First, transcription machinery unzips the DNA, temporarily separating the complementary strands. Second, RNA polymerase wraps around the DNA template strand. Third, RNA polymerase attached to the template strand. It will read the DNA to build a complementary strand of mRNA. Fourth, RNA polymerase reads the DNA template strands, adding building blocks to the mRNA strand according to the rules of complementary base pairing. G in DNA pairs with C in RNA. C pairs with G. T pairs with A. And A pairs with U. Fifth, RNA polymerase slides along the DNA template strand, unzipping the DNA and adding bases to the growing mRNA as it goes. Genes are typically thousands of bases long. So, we have just transcribed a small piece of an actual gene. Next step is translation. In summary is, the ribosome reads the bases of the mRNA, putting amino acids together to make a protein. The mRNA attached to the ribosome. The ribosome slides along the mRNA until it finds the bases AUG. AUG is the start signal for building a protein. It establishes the reading frame for building the protein. Transfer RNA or tRNA molecules attached to the three-lettered mRNA codons by complementary base pairing. At the other end, they carry an amino acid. So, at this point, we use the amino acid codon chart. We notice that AUG codes for methionine or M. You can see here M on the picture. The ribosome slides along the mRNA, moving 3 bases at a time. Inside the ribosome, each codon recruits a tRNA molecule, which brings in the next amino acid. The ribosome links the amino acid together to start building a protein. The ribosome continues along the mRNA molecule, reading codons and adding amino acids to the growing protein chain. When the ribosome reaches a stop codon, the mRNA and the finished protein are released. You can see on the picture here is the code UGA. It's meaning stop. So your protein is finished. Real proteins are often hundreds of amino acids long. The cell can read same mRNA strand again to build another protein. So we just transcribed and translated a very small piece of a real gene. So the gene that we just transcribed and translated came from corn. Corn, also called maize, is a plant that is farmed on a large scale around the world. It was domesticated thousands of years ago from a wild plant called teosinte, which grows in Mexico. Corn is used to make many edible and industrial products, including tortillas, corn syrup, animal feed, and ethanol. Here are some information about pCNA, or proliferating cell nuclear antigen, from corn. So they are MLELRLV GSLLKKVLEAIR. These are pCNA protein, partial amino acid sequence, 20 out of 275 in total. So the function, when 3 of these proteins come together, they form a ring that wraps around a single strand of DNA. The ring acts as a clamp for DNA polymerase, the molecular machine that copies DNA, so it can lock onto the DNA template strand during DNA replication. The structure: 3 pCNA proteins from the plant Arabidopsis thaliana are pictured to the right. DNA fits into the space in the center. The part of the protein that corresponds to the part you made is shown in darker shade. The overall shape of this group of protein is nearly identical between vertebrates, plants, and archaea. So again, transcription is a molecular machine or RNA polymerase attached to a gene and makes a messenger RNA or mRNA copy. Inside the nucleus, RNA polymerase attached to a gene. It separates the 2 complementary DNA strands, exposing the gene's unique sequence of bases. So A to U, T to A, C to G, and G to C. Remember, in transcription, A in DNA pairs with U in RNA. When the end of the gene is reached, transcription is complete. The new strain of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. And translation is the ribosome reads the bases of the mRNA, putting amino acids together to make a protein. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA or tRNA. Each tRNA molecule has its own anticodon and matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attached to the end of the mRNA strand. It slides along until it finds a start codon AUG through complementary base pairing. The start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein's instruction. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acid falls into its final form and the new protein is complete. This protein, amino acid tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life. Thank you for watching and listening.